So at this point, I'm going to hand the stage to Michael Yardson for the presentation or the introduction.
Yeah, sure. Yeah, I'll be super brief. Just to say it's a great pleasure to have Sam here with us, coming from one of the most beautiful places on earth. He's an assistant professor in Okinawa, Neuroscience Institute. Yes, you can see it and we'll be jealous. You know, we have a nice setting here. This is pretty amazing. And he also works on some of the coolest animals in the world. So he did his undergraduate in Brown, PhD in Brown and NIH, and then a postdoc with one of my scientific idols, Yudo One, at the Max Planck Institute in Frankfurt, where kind of, I guess this work also began developing. And along the road, I have to look at it because I work on so many different systems. So rat, fly, moth, locust, lizard, turtle, cuttlefish, octopus. So it's just this beautiful kind of way of studying systems from a computation perspective as well. And I think that's the title of your laboratory, Computational Neurotology, which is, I think, in this audience, like very near and dear to many of our hearts. And I just look forward to hearing about your super cool work. Hopefully, like, all the recent stuff and things that we don't know about yet. So very excited. Thank you for being here, Sam.
Thank you very much. Thank you for letting me just crash while I came over to the West Coast. Thank you for hosting, an invertebrate neuroscientist in the Vertebrate Museum. And thanks for coming to the talk. So today I'll tell you about octopus sleep. So to start, my work is really motivated by a fascination with the diversity and complexity of animal behavior. So every day, animals are they're faced with the challenge of surviving in hostile environments where they only have limited and noisy information coming in through their senses and only a limited amount of time to learn from their mistakes. And yet they can thrive in these environments. And I think often we don't know how they do this. And I think that this is likely because there's general principles of animal brains and behavior that we don't currently understand. And it's in searching for these sort of general principles that led me to start working on the cephalopods. So these are the coleoid cephalopods, cuttlefish, squid, and octopus. And these are marine invertebrates. They're mollusks, and they have independently evolved really large brains and complex behavior. So independently from our own vertebrate lineage. So the high-level approach at the lab is to try to compare between these really different animals and more familiar animals like vertebrates. And if we could find any commonalities across such different groups, I think this points us towards truly general principles of brains and behavior. And we can use this to separate out what's general from what's species specific or clade specific. So one of the amazing behaviors the cephalopod can do is camouflage. So they have the best camouflage in the animal kingdom by far. This is an octopus we filmed nearby Oist in Okinawa. So they can change their color and their shape and their texture at a moment's notice in order to help them avoid detection from their predators and also to help them hunt for their prey. So if we zoom in on an octopus, this works for all the cephalopods. You can see that the camouflage is actually composed of tens of thousands up to millions of these special skin cells called chromatophores. And the chromatophores come in a range of different colors, and they can be expanded and contracted under control of these animals' brains. So how it works is that every chromatophore, here's one, is lined radially by a series of muscles. So here it's an actin stain, so you can see the muscles. And motor neurons project all the way from these animals' brains. So they have the big brains right between their eyes. And the motor neurons project all the way down to the skin and synapse onto the muscles lining small collections of chromatophores. So each motor neuron synapses on between 1 and 10 chromatophores, and each chromatophore receives input from between 1 and 3 motor neurons. And so when the motor neuron is active, so it's firing action potentials, it contracts the muscles that line every chromatophore and expands the chromatophore. And when the action potentials stop, the chromatophore contracts back down And you can see the white skin underneath the chromatophore in the skin. And so it's because mechanistically, this is how camouflage is generated under nervous system control. That I think if you could record the expansion state of all the chromatophores, it would give not only a really interesting quantitative description of this camouflage behavior, which is so fascinating, but also you'd be indirectly measuring the activity level of the large populations of motor neurons that's causing the chromatophores to expand and contract. So over the last few years, we've been working on this. So the techniques for recording large sets of chromatophores in freely moving animals and trying to infer the motor control in this way. But I'm going to tell you today about sleep, and I think pretty quickly you'll see how these things relate. So when you went to sleep last night, your brain alternated between two stages. So there's REM sleep, where you have these rapid eye movements, and your brain paradoxically looks like it's awake. And then there's slow wave sleep, where there's large, low frequency oscillations throughout the cortex. There's many different ideas of why we have two stages of sleep, why we alternate between them as we do. But I think it's safe to say that the question, why do we have two stages of sleep, is not clear. The answer is not clear. Lots of ideas, some of them mutually exclusive. It's a mess. But it's in this context, so two stages of sleep was identified originally in humans in the 50s, and then been studied profitably in mammals. So it's found in every mammal where it's been looked for. Recently, though, it's two stages of sleep had been identified in other vertebrates. So I worked in my postdoc. We identified two stages of sleep in a lizard, so reptile. It had previously been shown in birds, subsequently in fish. So a parsimonious explanation for this is that two stages of sleep evolved very early in vertebrate evolution, and it's been maintained over hundreds of millions of years and in such diverse animal groups because it's doing something really important, but we don't know what it is. So in this context, there's been a couple recent reports of something like two stages of sleep in octopus. And this is, I think, really interesting because octopus diverged from the cephalopods, diverged from the vertebrate lineage about 550 million years ago. So our common ancestor was a small marine worm that didn't have a very complicated nervous system at all, presumably did not have two stages of sleep. We don't see two stages of sleep in other invertebrates. People have looked in Drosophila, C. elegans, jellyfish. So if we could observe two stages of sleep in octopus, it would perhaps be an example of convergent evolution of this phenomenon, speaking to its general importance. So when I came to OIST a number of years ago now, it was the winter, which is octopus season. So we could go out at low tide and collect small octopuses from the tidal pools. It's really hard. They're active at night. It's really hard to catch them. Their camouflage is very good, but also they sit on top of their dens, and they can dive down into their den through just a few, maybe a centimeter wide opening, and you can't get them when they dive down. But with some practice, we can catch them and bring them back to the lab. This species, it turned out to be a local species, Octopus laqueus. They will happily sleep all day in the lab in flower pots. This is what a sleeping octopus looks like. So here's the eye, it's closed. The arms are tucked up underneath the animal. And the animal's all white because the motor neurons that would cause the expansion of the chromatophores are not active. So periodically, we have observed this. So this is a real-time video of the sleeping octopus. So you can see the eye movements, the rapid eye movements, the skin and body twitches, and rapid transition through a series of skin patterns. And this behavior, which we call active rest, lasts for about a minute before the animal settles back down into its normal, we'll call it quiet sleep. So to quantify this behavior simply, we trained a neural network to identify the octopus in our scene, and they can do a good job of this. And then we could just simply take the average brightness of all the pixels that the network says is an octopus. So this reduces the image down to a single number. So this is the video I just showed you, but now just looking at the average brightness of the octopus starts off white in quiet sleep and then goes through this series of darkenings and lightenings for this active rest, and then it settles back down to the normal white color of quiet sleep. And there's these periodic, really kind of subtle to see by eye flashes of color. This happens throughout the sleep, and we're not quite sure what it is. But if we keep our camera rolling, we can observe that this active rest behavior repeats. So every about an hour, you have a minute of active rest, and then the quiet sleep in between. And this happens all day long, and then at night, the animals wake up and hunt for crabs. So it seems that this rhythmic occurrence of active sleep is under circadian control. So we can record continuously for many days, and normally there's a 12-hour light dark cycle. But if we keep the animals show a lot of these active rest bouts during the day and very few at night when they're moving around. If we keep the lights on for 3 days continuously, we still see a roughly 24-hour cycle to this behavior, and it's entrained to the onset of a normal light dark cycle again. And similar results when we keep the lights off for 3 days. This behavior is also under temperature control. So these animals don't regulate their body temperature. So we can simply change the temperature of the water to change their brain temperature. So on the x-axis here is varying the temperature, and the y-axis is looking at the time between active rest bouts. And it seems that every degree that the water gets warmer, the time between these active rest bouts shrinks by about 8 minutes. And it's roughly linear. And besides looking at the color of these animals, we can track points on the body and with optic flow and show that relative to their normal quiet sleep, this active rest behavior comes with more eye movements, more body movements, the breathing rate goes up, and also the variability of the breathing. So is this active rest behavior sleep? Is this a distinct stage of sleep? So to test this question, we relied on the evolutionarily conserved behavioral criteria for sleep. So across animals, from jellyfish to humans, there's a set of behaviors that are conserved. And so we could test whether active rest meets those criteria. The first of those criteria is whether this behavior is rapidly reversible. So this separates sleep from things like death and coma. You can wake the animal up. So we attached a electromagnet to the side of the tank, so we can give a mechanical tap at controlled strengths. So this is a picture of the octopus sleeping a second before we tap it. This is a second after we tap it. It does a startle response and changes its skin pattern and color. And we can track the movement with optic flow. So this is when we tap. So we can see the movement. So if we give a strong tap here, whether the octopus is awake in quiet sleep or in this active rest stage, the octopus always reacts. So it's rapidly reversible. We then dialed down the strength of our mechanical tap about 80-fold. And here we can observe a regime where the octopus will react when it's awake, but not when it's in active rest or quietly sleeping. So active rest comes with a heightened arousal threshold, which is another criteria for sleep. And the third criteria is whether this behavior is under homeostatic control. So again, the octopus is sleeping and resting all day. it's active at night. We can track it at night under red light, which they can't see. So we can track it here for 36 hours. At night, there's very few active rest bouts because the animal is active looking for crabs. There's a lot of active rest bouts during the day. Again, at night, very few. We then deprive these animals of active rest by tickling it with a paintbrush continuously for 2 days. And then my grad student, I think he did it. And then we let them freely behave and see what they do. And we observed that night, after the days of deprivation, the octopus do more active rest bouts. The following day, there's just as many as before the deprivation. But 2 days later, again, there's more active rest bouts. So this behavior is rapidly reversible, and it comes with a heightened arousal threshold, and it's under homeostatic control. So passes all the criteria. I think we could talk about this as being a stage of octopus sleep, active sleep, and this the normal quiet sleep, so 2-stage sleep in octopus. Sorry. We tested how fine the homeostatic control is. The experiment I just showed you earlier was we were just continuously depriving the animals of both active and quiet, active rest and quiet sleep. Here we selectively disrupted this active rest stage. So we waited for the animals to start active rest, and then we bothered it with the paintbrush. And you can, if you do it right, they don't wake up. So we can then count the amount of quiet sleep before the next active bout. And what we observed is that when on a single bout resolution, when that bout was disturbed, the next active rest bout happened not the normal hour later, but about a half hour later. So it seems that the octopus are really going out of their way to get their active rest in. So it's really finely under homeostatic control. So anyway, these are 2 stages of sleep in octopus. So we're interested in what's happening in the brain when the animals do this sleep stage, these 2 stages of sleep. Recording from the octopus brain is really difficult because there's no skull, and the octopus is underwater and it's squishy. But it turns out that the octopus brain is surrounded by a cartilage casing that's it's not a skull, but it's firm enough that you can head fix to. So we head fix the octopus, and this allows us to put neural probes into the brain. These are neuropixel probes. They're multi-site silicon probes that we can record many locations of the brain at the same time. And luckily, the animals will fall asleep and do normal sleep behavior under head fixation if we do the experiments in the morning. So here's looking at this rhythmic occurrence of active sleep bouts in freely behaving animals, where every hour you have about a minute of active sleep. And then when we have it under head fixation, again, we can see this every hour. There's about a minute of active sleep. So this lets us study what's happening in the brain during this interesting behavior. So when we put our electrodes in, this is the transition. This in red is the skin pattern color. So this is the quiet sleep to active sleep transition. This is in black, a single local field potential. So some electrical activity around a region of the octopus brain. As it transitions from quiet sleep to active sleep, we can see that the brain really explodes with activity around the onset of active sleep. Here's a spectrogram showing the frequency content. It's mostly show here. Yeah, there's relative to waking power, the active sleep comes with a heightened low frequencies as well as about a 30 hertz oscillation. Compared to that quiet sleep, the brain really looks like it turns off. Everything across frequencies, the power is dampened. So we don't observe slow, large oscillations similar to slow wave sleep in vertebrates. When we saw this sort of neural response, we wanted to know where in the octopus brain this is happening. So to localize our signals, we took a tissue clearing and light sheet imaging approach. So what you can't see here is the octopus brain because we've turned it transparent chemically. It's a bit yellow because we have a nuclear stain. We built a light sheet microscope to image these brains in 3D at a very high resolution. So the brains are about 2/3 of the size of a mouse brain, if you know what that looks like. This is the octopus brain in 3D coming from our scans. So yeah, if you know what a vertebrate brain looks like or an insect brain, it doesn't look like this. Like it's its own independently evolved structure, and it's almost completely unstudied. I think it's really beautiful. So for this study, we're recording from the central brain. There's these two optic lobe structures, these pills on either side, do visual processing. We couldn't study them for this study, but we're working on these now, how the visual system works in these animals. So we could segment different regions of the central brain using some classic anatomical work as guides. So we have a number of brain regions. This is a 3D atlas of the octopus brain. And then we can take our neural pixel experiments. We coat the electrode with dye so we can localize it anatomically. We can map these sample brains into our reference brain using non-rigid deformation. So we can build, this can be done pretty well. So here's in red is a high cross correlation between the reference and sample brains. So this lets us take all the results from many experiments and map them all into a single reference. Here's the octopus brain in gray. Each line is the location of a neuropixel probe, and there's electrodes all the way up this probe. I've colored the electrode according to the average activity level during active sleep. So in this frequency band. So you can see every time we put the neuropixel probe through certain brain regions like this one, the superior frontal lobe, all the electrodes in that brain region show a lot of activity. So this example I showed you before came from this region. Other brain regions also show activity during active sleep with a somewhat different character. So this large brain region up here called the vertical lobe is also very active during active sleep, but it has this low frequency, strong deflections. It looks quite different than the frontal lobe. But for this study, we were interested in comparing neural activity during active sleep and during wakefulness. So we could get waking data before the animal falls asleep in our head fixation. Sometimes they wake up in our experiments, so we can compare. So we looked across different brain regions here on the x-axis, and we did it. We looked at low frequencies as well as high frequencies, and we looked at all the electrodes for a brain region. This is the violin plots, both so comparing active sleep, wakefulness, and quiet sleep. So we're looking pretty systematically. There's in general a high correlation between the neural activity of a brain region during active sleep and that of during wakefulness. And in contrast, in quiet sleep, everything looks pretty silent, with a few exceptions that I'll get into. So it seems that that active sleep comes with wake-like neural activity. So quiet sleep, in contrast, everything turns off, except in certain brain regions, on top of this pretty silent background, there's these, what we call oscillatory bursts. There's these short, about a second long deflections in the local field potential. So here's 4 of them. They're at about 15 hertz. So here's a zoom in on one. So it takes about a second. There's several cycles of this oscillation. And if we look for these everywhere in the brain by just taking a big fantastic filter, we can find thousands of these events. They all look more or less similar. And they're localized in specifically, I think I can show it here, this frontal lobe region and the anterior part of this vertical lobe region. So these two brain regions are interesting. They're directly connected anatomically. And when you remove them from the octopus, they lose spatial memory. So they forget the location of crabs. So it seems that selectively in these sort of learning and memory centers, there's this these oscillatory bursts that resemble a phenomenon in human sleep called sleep spindles. So it resembles sleep spindles both in the duration of these bursts and also the frequency content of the LFP. So it was pretty striking to find. So lastly, we looked at the content of active sleep. So what are these patterns that the animal is transitioning through so rapidly? So we can film the animals from the top, so we can see a complete view of the skin, and we recorded about 100 of these active sleep bouts at very high resolution. And so previously we had been describing the skin patterns just by taking the average intensity of the skin, so the average brightness. That's shown here in black. This is not a really good measure of this behavior, because you can have examples like this pattern, which has this average brightness, and this pattern, which has a very similar average brightness, but you see that the patterns are quite different. We need like a higher dimensional, richer description of this sort of thing. I wanted to develop something that could pull out something interesting that I saw by eye, where in a certain active sleep bout, the animals will do a series of patterns. So let's take this pattern as pattern 1, this pattern is pattern 2. If I look an hour later at the next active sleep bout, at least by eye, I can see the same sort of patterns show up again. And the next active sleep bout, I see the same patterns show up again. So I wanted a quantitative description that could pull this sort of observation out. And so what we settled on was a pipeline like this. First, I segmented out the mantle of the octopus, and I can align these mantle images and to produce a standardized image. And I could feed these images into a deep neural network. This is the VGG19 neural network. It's pre-trained on ImageNet, which is a large corpus of natural images. And previous work had shown that the representation of neurons deep in this network form a useful parameterization of visual textures. And this is used in machine learning for things like style transfer. And so a reason that we have a visual texture here on the animal's back. So we could use this representation as a higher dimensional. So there's 512 neurons in this fifth layer of this network. So it's a 512 dimensional representation of the skin pattern dynamics during active sleep. So with this way of describing it, we could take those 512 dimensional dynamics. Here it's looking at the top 2 dimensions, the top 2 principal components. This is what one active sleep bout looks like projected into this space. Here the time is coded on in the rainbow. And every 10 seconds, about from this minute-long active sleep bout, I pulled out one pattern, so you can kind of see what this looks like. So the animal starts active sleep here, it's all white, and then it transitions out here to this dark pattern, stays dark, even darker here, and then it shoots back to here, which is white again. Then it goes out to this green pattern, which I don't know how to describe that in words. And it goes through this sequence of patterns. So now that we could describe this quantitatively, we asked, so what's the closest pattern to any given pattern in this bout in a different bout? So how do these patterns repeat quantitatively? And it seems that they do, at least described in this neural network. So here's for this pattern, what's the closest pattern in a different bout from this octopus or from other octopuses? You can see that the columns here look pretty similar, at least macroscopically. So it seems like in different active sleep bouts of an individual and also across octopuses, there's a conserved set of patterns that are reappearing. Oh, sorry. There. So just to look at the dynamics a little bit more, we calculated a few simple statistics. So the first was just looking at the how far away or subsequent images in an active sleep bout. This produces this blue histogram of distances, so they're close together in our high frame rate camera. We then looked across multiple bouts, or taking pairs of bouts, starting at the beginning of the bout, seeing how far away do these two trajectories get over time. We look at the average distance over all times, and that's this distribution way out here. So it's very far compared to this one. So it looks like the dynamics of an active sleep bout are not repeating. They're not going through the same sequence of patterns in the same order every bout. It differs. But if we ignore time and just say, how close does a pattern in one bout get to any other pattern in another bout? And we have this distribution of distances that more approximates that of the next point along a trajectory. So it seems here that every active sleep bout, the animal is taking a walk over its pattern repertoire. It's a different walk every bout, but it's over a conserved set of patterns. So what are these patterns? It's not working there. Sometimes we could observe the patterns of the octopus when it's awake, and sometimes it's laid out nicely that we can see it. We can find the matching patterns when we look at active sleep. So here's two patterns, and I can lay these images on top of each other and warp them. This is showing the overlap. So purple is the waking pattern, green is the sleeping pattern, white is the overlap. So these patterns really strikingly resemble each other. Here's another example. This is we call the polka dot pattern. The animal does this when they're annoyed. So when you kind of put your hand close, they'll do this pattern. You see this pattern show up in active sleep. It's darker, so every chromatophore is more expanded. But if you look at the structure of these light circles here, and this you can see better once it's been aligned, it's really strikingly similar. So it seems that in active sleep, it's a walk over their waking pattern repertoire, and the waking patterns are reactivated during octopus sleep. So to summarize, I've told you now how octopus have two stages of sleep, quiet and active. Active sleep resembles REM sleep in vertebrates. Behaviorally, it's also accompanied by wake-like neural activity. Quiet sleep is accompanied by the sleep spindle-like events in known memory centers of octopus. Wake-like skin patterns reappear during active sleep. And I hope I convinced you that the skin patterns in these animals provide a really unique access into the inner workings of the brain, both in wakefulness and now in sleep. The range of similarities that we can observe between vertebrates REM sleep and octopus active sleep suggests to me that we're looking at the convergent evolution of similar phenomenon. It's possible that large brains, complex behavior, and an active wake-like sleep stage all go together. Why? I'm not sure. But I think it's really striking that evolution would hit upon such a range of similar phenomenon twice, at least. So just a teaser, we're also working on, we're starting to work on cephalopod vision. So cephalopods have independently evolved eyes that work just like ours, camera-type eyes. But instead of our retina that does a lot of pre-processing, the cephalopod sends photoreceptors directly to its brain, and there's a retinal structure. And then after that, the visual center of the cephalopod looks nothing like any other visual system that I know of. It's weird tree-like thing, and these are the neural processes. Somehow they see with it, and behaviorally they're very good at vision. It's a total mystery how this works, so that's exciting to be working on. Let's skip this, sorry. Okay, with that, I'd like to thank the members of my lab, especially Aditi, Kazumichi, and Tomo, who worked on this octopus project. We're working with Leno and Mishralam at University of Washington. And yeah, thank you for your attention. Happy to take any questions.